Good morning, good afternoon, and good evening, everyone, and welcome to our webinar series, New Tools of the Trade in Life Sciences, Technology that Enable Breakthroughs. Today's webinar will focus on isolation and characterization of endogenous nuclear pore complexes. My name is Renate Weitzbauer. I'm the lead editor of the webinar series at Bioprotocol, an open access online journal whose mission is to make research more transparent and reproducible. All right. It is now my distinct pleasure to introduce the moderator of today's webinar, Dr. Samson Obado. Sam is currently a research associate in the Laboratory of Cellular and Structural Biology at the Rockefeller University, where he has worked extensively on the nuclear biology of trypanosomes, which cause public health problems in most of the developing world and has also spread more recently in the United States. Sam completed his PhD at the London School of Hygiene and Tropical Medicine, where he focused on chromosome biology and cell division in trypanosomes. He then moved to the Rockefeller University in New York, where he's been working on the evolution, structure, and function of the trypanosome nuclear pore complex, including nucleocytoplasmic transport of proteins and RNA. And with that, Sam, thank you so much for being here today. And the floor is all yours.
Thank you very much. Welcome everybody to today's webinar. I hope you're excited as I am to hear what Javi has to say. First, a quick few words about Javi, who I've known for quite a long time now. Javi did his PhD at the Biological Research Center in Madrid, where he was studying the regulation of nuclear transport of a transcription factor involved in environmental pH sensing. And then for his postdoc, he joined the Rout Lab, the Laboratory of Cellular and Structural Biology at the Rockefeller University under the supervision of Professor Mike Rout, where he studied the structure, function, and evolution of the nuclear pore complex, which is the main regulator of nuclear transport. He recently moved back to Spain, to Bilbao, where he has started his own lab as an ICABASC research associate and group leader at the Biophysics Institute. And with that, it's my pleasure to give Javi the floor so he can tell us his story today.
All right. Thank you, Sam. First of all, I would like to thank Sam and John for helping here, and especially big thank you to Bioprotocol and especially to Cindy for organizing this whole series of seminars. Let me start presentation. All right. So the work I'm presenting today, it's not just my work, it's a fruit of a massive collaboration between a big consortium of laboratories, amazing laboratories and scientists that have been working on all this, and that I will acknowledge as I go through the story and at the end. And today I'm going to talk to you about the isolation and characterization of a fascinating molecular machine that lives at the heart of our cells, and that is the nuclear pore complex. And our story starts at the nucleus. It is the quintessential eukaryotic organelle. It's a round-shaped organelle formed by a double lipid membrane called the nuclear envelope that surrounds and encloses the chromatin and separates our genome, or the genome of the eukaryotic cell from the rest of the cell. The presence of the nucleus basically physically compartmentalizes the genome, separating many different activities, and that is thought to have provided eukaryotes with a whole new level of regulation that was key in their evolutionary success. But the presence of this nuclear envelope, like a plastic bag around the genome, also poses a problem, and that is a communication problem. You have to be able to communicate the inside of the nucleus with the rest of the cell. And eukaryotic cells manage to do that by developing massive, from a molecular point of view, channels that generate a connection between the nucleus and the cytoplasm, and actually establish what we can consider a virtual gate. These channels are called nuclear pore complexes, or NPCs, and they are the true gatekeepers of the nucleus. The NPCs are large, are huge. It's a massive channel. Here you have a comparison of an ion channel with a ribosome, and this is the size at scale of an NPC. In yeast, it has around 50 megadalton. It's even bigger in mammalian cells. It will go to 120 megadaltons. And is formed by more than 550 copies of 30 different proteins called nucleoporins. The NPC is the main mediator transport between the nucleus and the cytoplasm. All macromolecules and molecules that go in between the nucleus and the cytoplasm have to go at some point through the nuclear pore complex. And it's an 8-fold symmetrical assembly that is formed by 8 protomers that we call spokes that connect radially. Here you have an example of this spoke, 8 of them connecting radially, and they form also what we call concentric rings. And for following what I'm going to talk about, you just need to know that there's one of the rings is the membrane ring, that is proteins directly associated to the nuclear envelope membrane, an inner ring that forms the core scaffold, and sandwiching in between this, the membrane in the inner ring, are the 2 outer rings in the NPC. This arrangement of spokes and rings form a channel, a 30 to 50 nanometer channel that that traverses the nuclear envelope. But this channel is not empty. It is filled by a special type of feature that we call the central transporter. And the central transporter, located here in the central channel of the NPC, it's actually also mainly formed by a special type of nucleoporin that are rich in phenylalanine glycine repeats and are also intrinsically disordered. These phenylalanine glycine rich repeat proteins are called FGNAPs, and they are the ones that wiggling and filling the central channel form the central transporter and the permeability barrier that regulates transport. So basically, these proteins are not going to allow the access of things that should not be entering or exiting the nucleus. They allow the passive diffusion of things that are below a cutoff of 40 to 50 kilodalton. Things that are over that size require an active transport to get in or out of the nucleus through the mediation of specific transfer factors. And this type of transport, nuclear transport, is actually a very crowded environment because nuclear transport is actually really, really fast. Each transport event where a transfer factor takes a protein and gets it through the nuclear pore complex, it takes a matter of milliseconds. And you can have close to 1,000 of these events per second per NPC. So this molecular machine is able to deal with a massive and constant flux, yet at the same time being able to regulate that flux into what is specific and what is not. So it's a really remarkable system. But apart from its main role as mediator of transport in the nucleus, the NPC is also a key regulatory platform for many essential nuclear processes, like, for example, RNA maturation and quality control, DNA damage repair, transcriptional control, chromatin architecture, many different aspects of nuclear biology. And underscoring this importance as a regulatory role of the NPC is the fact that many mutations affecting components of the nuclear transfer machinery or components of the nuclear pore complex have been shown to be directly related to many human diseases, like, for example, several types of leukemias, cancers, and several neurodegenerative diseases. However, the basic molecular mechanisms that underlie these regulatory roles of the NPC are actually still poorly understood. And if we wanted to understand how the NPC works both in health and disease, what we needed to start with is a proper framework, is a detailed description of the structure of how an NPC is assembled. However, the structural characterization of the NPC has been traditionally very challenging because, well, it's a huge complex, is very large, is very complex, formed by many different components. And also the fact that nearly 1/3 of its components are predicted to be disordered or intrinsically disordered. So when we approached this problem, we thought that no single technique is going to be able to address all those different challenges. And we decided to use what we call an integrative approach or hybrid approach for the structural determination of the NPC. An integrative approach is sort of analogous to the solving a structure through nuclear magnetic resonance. In NMR, you will have a signal for pairs of atoms. The signal is then translated into spatial restraint, and all those restraints are then used to calculate an ensemble of a structure that fits the data that we fed this calculation. In an integrative approach, we have something similar, but instead of pairs of atoms, we have other type of we have proteins, we have domains, we have crystal structures, and it's a 4-step iterative method that starts with basically the first step is gathering experimental data. And this experimental data, it's ideally coming from complementary approaches, complementary methods that are going to inform us about different aspects of the structure. Ideally, also, these different methods should cover a wide range of resolutions so that we can maximize the synergies between the data and the final accuracy and completeness of the final structure. So again, we gather a very diverse set of experimental data. This data is then translated into spatial restraints that are used by the integrated modeling platform developed by the laboratory of Andrei Sali at UCSF to calculate an ensemble of a structure by satisfaction of all those restraints that satisfy the input experimental data. So this is a calculation of a structure. We are not modeling anything. Finally, the final step is the analysis and validation of the structure and deciding if we need to improve certain parts for which we can go back and collect new data. So to apply this approach to the nuclear pore complex, the first thing that we needed to do, again, first step is to collect experimental data. And to collect this type of experimental data, we needed to put our hands in those NPCs. We needed to be able to get isolated NPCs. And to do that, there was already a method, and I will explain next. And we decided to use a model organism. In this case, our model organism is the yeast, the baker's yeast, Saccharomyces cerevisiae. This is a very widely used model organism that in our hands and for our purposes has several very interesting advantages. First of all, Saccharomyces, it's a simple unicellular eukaryote that can be easily grown, is quite inexpensive to grow, and it can produce really high yields. Next is it's all the basic molecular mechanisms in a eukaryote are conserved, highly conserved in Saccharomyces cerevisiae, and that includes most of the transfer pathways and the structure of the nuclear complex. Then Saccharomyces has very powerful genetic tools that will be very interesting to combine with our biochemical and structural biology methods. And finally, Saccharomyces cerevisiae lacks a well-organized nuclear lamina. What Saccharomyces cerevisiae has is a quite kind of a diffuse lamina-like arrangement of these two proteins, MLP1 and MLP2. So the nucleopore complexes are not really connected through a very stable and resilient network like the lamina, and that helps in the isolation of these NPCs. So the first method or the most successful method developed to generate and isolate nucleopore complexes was developed by Michael Rout in Gunther Blobel's lab. And it was a method that was really awesome. It allowed us to purify nucleopore complexes very successfully, but it has some disadvantages that we needed to address. Basically, as you can see here, it's a long and complicated protocol. Some of these fractionation steps, they have to be run overnight, so it takes several days. It required a significant amount of starting material, and also it was really efficient only in one specific yeast genetic background. So to try to develop a better method that would be more suitable for the experimental data gathering that we wanted to do, in collaboration with Ilona Nudelman, a postdoc, a very talented postdoc in Mike's lab, we developed a new affinity method that, in comparison with the previous method, allowed us to basically purify these NPCs really fast. Compared to days, we are able to get these NPCs in a matter of a few hours. Is very efficient. We don't need huge amounts of material to obtain these NPCs, and on top of that is basically virtually applicable to any yeast genetic strain or genetic background that we can imagine. So with all those advantages in hand, we decided this was the method to use. And this affinity purification method is actually not very complicated, although the different steps took a lot of time and effort to really optimize to the point that we were getting native, intact, and high quality nucleopore complexes. The method basically starts with, and it's analogous to basically an affinity purification method for nearly any protein complex that you can imagine. So yeah, it will start with your favorite cell. And because we want to preserve these complexes in their native state, we freeze these cells under liquid nitrogen conditions. And under the same liquid nitrogen condition temperature, we lyse the cells. And we managed to do that using planetary ball mills that are cooled down under liquid nitrogen conditions. And you can find several protocols at the NCDIR protocols website. I'll talk about that later a little bit more. But basically, using these planetary ball mills and cryo mills, we can break the cells under liquid nitrogen conditions, preserve the complexes in their native state, and we generate what we call cell powder. Is a fine whitish powder, frozen, that can be stored at -80 for a very long time, and they're completely preserved. After that, with the powder, what we do is basically we quickly resuspend it in a liquid buffer. These buffers also have been previously optimized to get the best conditions possible to serve several purposes. First of all, to keep our complex intact and to prevent bad things to happen. I'll explain that in a little bit. But basically, this step, I have to underscore that it's very important, the optimization of the buffer conditions. And I would like to point you to a very interesting paper by Sana Hakberdian and John Lacava, where you can actually get really high throughput analysis of many different buffer compositions. So you can explore the buffer universe to optimize it to the purification isolation of your specific complex. In the case of the NPC, we use this method and other not so high throughput methods to optimize the buffer conditions. In those buffer conditions, we use paramagnetic Dynabeads coated with a specific antibody to a tag that we have in our nucleopore complexes. We bind them to those Dynabeads, wash the non-specific components of the cell milieu, and then through protease cleavage site that we engineer in between the tag and the protein that we're using as a handle, we can elude under native conditions our complexes. This kind of native elution can be also done using different methods. In our case, again, the one that worked really well was to cleave using specific protease, also is the human rhinovirus 3CA protease that I think is commercially called precision protease. That has also several advantages. It's very specific, and it also can cleave at 4 degrees of temperature. So again, we keep our complexes cold and happy and preserved during the whole process. Finally, once we have our complexes, we can then analyze them, test that they are exactly what we want, and next we can go downstream to the downstream analysis and handling of these complexes to perform all the data gathering that we wanted to do. And in terms of this protocol that actually you have in the method paper that I refer in the previous slide, I would like to stop a little bit in several aspects. Because, well, again, when we have our cells and we break them apart, you slice the cell, the only thing that is going to happen is bad things for your complexes. And this kind of bad things that start to happen are basically degradation, disassembly, and non-specific interactions. Degradation because you break the compartmentalization of the cell. So now everything is mixed together, including proteases, phosphatases, many different enzymes that should not be usually in contact with the nucleopore complex or with your complex are now going to get in contact with your complex of interest. And you have to try to avoid the action of those guys over your complex. That is going to be usually degradation, dephosphorylation, and things that you don't want to happen to maintain the native state of your complex. Then the other bad thing that is going to be starting to happen is disassembly. Obviously, these complexes, once you break the cell, they are extracted from the native environment, and especially labile or loosely bound components are going to start to fall apart. So you have to take that into account and try to use methods to prevent this kind of stuff. And the last thing that is going to start happening when you break the cell apart is that abundant and sticky proteins are going to start to non-specifically associate with your complexes, especially when the extraction of the complex may expose hydrophobic patches, and these regions of the complexes are usually very attractive to this type of sticky proteins or heat shock proteins. They are going to start adding non-specific interactions to your complex. So the best way or the best practice is to try to prevent all these deleterious effects are basically 3 of them. I mean, how to minimize this is work really fast. So speed is important. How fast you perform your affinity purifications. Affinity purifications that run overnight, as I used to do many, many years ago, to be honest, are not great. All these processes are going to be at their best in those long time purifications. Second, try to keep the temperature always low. And another option to prevent especially degradation is the use of inhibitors, protease inhibitors, phosphatase inhibitors, any type of chemical that can prevent some of these effects from happening. It's usually something interesting to test, to try in your complex. And a little bit more specific of the nucleopore complex, at least in our hands, are several key steps that I would like to mention. Is also the type of affinity purification matrix. There are dozens of them in the market with different properties, different conjugations, cepheros, agarose, you name it. They all have different properties, but in our hands, the affinity matrix that usually outperforms all the others are Dynabeads. So I say here, we are not sponsored by them, but it's just my honest recollection after many years of doing this. Dynabeads are usually the ones that work the best because it's a solid matrix. They don't have, it's not porous. So there's no size exclusion limit to what you can pull out. They come in very convenient formats. They're super homogeneous. You can look at that just by putting a little bit of them under a microscope. They are very versatile in the type of things that you can conjugate to them. And in general, they also perform pretty well in terms of non-specific background. So that's what we usually use and what we will recommend in principle. Then, especially for the nucleopore complex or complexes that are really large and big, in our experience, you have to give them a little bit of love. You have to be gentle. This very big complexes, when you think about it, at the nanoscale, still they are subjected to several type of forces. And for example, as you can see here, when you are mixing them with the Dynabeads, you're going to have shearing forces, impact forces that can potentially break down and degrade your complex. So in our hands, we have seen that, especially for the nucleopore complex, you have to be as gentle as possible. No room for, I don't know, heavy vortexing or stuff like that. That's only going to cause problems in your samples. Next of all, and I said it already, is the buffer composition. And that's on itself a whole universe, and we could have several webinars talking about buffer compositions and what would be the best. There's no one that is the best, is which one is the best for your complex, which one is going to preserve your complex intact and in the best conditions, and potentially also with preserving the activity that they may have in vivo. And not necessarily these buffers have to be what they call physiological. Once the cell is broken, there's nothing physiological anymore. So again, you need to tailor your buffer composition so that your complex is happy, intact, and stable. In that case, again, you can go to some of the literature that I mentioned before, and you can see that there are also many components that you can use. Some of them will help enhance the stability of your complex, like cosmotropes or glycerol. Some of them will be useful in preventing non-specific interactions or aggregation between the complexes, like certain type of surfactants and detergents. You also need to extract your complexes, sometimes from membranes, sometimes from other cellular compartments. You need detergents, you need certain type of salts. And again, you can also add inhibitors to prevent degradation and to prevent your complex from being damaged. So again, we have very specific ones for the nucleopore complex. That doesn't mean that it's the best. It's the best of the ones that we've tested. Again, there's probably a whole universe of buffers that would be suitable for doing this type of protocol. And you always have to, I'm sorry, it is the way it is, but you have to spend time and effort in optimizing your buffer composition to get good extraction of your complexes. And finally, something that we've also found was potentially important is the localization and suitability of the tag protein, of the handle protein that you're going to use to pull from your complex. In our hands, we have used the tag that we usually use is protein A. proteinate from Staphylococcus aureus, because it allows us to use then in the matrix just rabbit IgG, what is quite cheap. But that doesn't mean that you cannot use any other. If you have an antibody, a good antibody that is specific to your protein, go for it. It should work very nicely. In our experience, what we have found is that the ideal handle is usually one that is located more peripherally in the periphery of your complex, not inside, not in the core scaffold of your complex, because that way you facilitate the access of the matrix to those tags, and you prevent strike entrances and other issues that will cause for you to purify stuff, but usually it might be broken complexes or degraded complexes and not really the intact native whole nucleopore complex in this case. So once you have this nucleopore complexes isolated, you can use different readouts to check the that what you're getting is an optimal prep. And what we have usually used is some of them are quite simple, like SDS page, as you can see here. This is how a pure NPC prep looks like in an SDS page. The second one that is not so straightforward, but I think that most people now have access to this type of methods, is mass spectrometry, where you can analyze what are the protein composition of your sample. In this case, as you can see, we can easily identify every single of the nucleoporeins that form the NPC. And importantly, because we are preparing these nucleopores really fast and in a very optimized way, we are actually also identifying and preserving most of the transfer factors and flux that was going through those NPCs at the point of isolation. So these factors, although being labeled, because we are being really fast and very efficient, we can preserve them and they co-purify with our samples. Another very interesting way of looking for the quality of your sample is negative stain electron microscopy. In this case, as you can see here, these are several views of one of our preps. You can see here beautiful nucleopore complexes, even with the central transporter nicely preserved. And negative stain is quick and relatively quick and easy way of checking that your prep is intact, your NPCs look good, and that you have good densities depending on your downstream application. And another method that is also quite interesting, sometimes a little bit more challenging, would be cryo-electron microscopy, because here we have to use a stain, usually a uranium salt, that could also cause certain type of artifacts depending on your complex. So cryo-electron microscopy of this same type of sample looks a little bit like this. This is raw pictures from cryo-electron microscopy, and you can see all these disc-like shapes are nucleopore complexes. You can even see the central transporter in some of them. And ideally, especially for cryo, you should also see that you have been able to freeze them in different orientations. These are all side views of the nucleopore complex. Using all these different readouts, you can then decide if your sample, if your prep is good enough to then proceed to analyze them through other methods. And for the structural characterization of the NPC, that was what we were interested in. The type of data that we needed to gather is the type of data that you would need to build any structure. And what is this type of data? The type of data you need is, first of all, you need to know which are the components and the copy numbers of your assembly. Next, you need to know how they are connected to each other, the connectivity. Finally, you need to know the morphology of the different parts and of the whole complex. And once you have all these different type of information, you can finally build the final structure. So I'm going to briefly tell you what type of data, how we collected all this type of data for the nucleopore complex, starting with the components and copy number of all of them. We used a combination of different techniques, starting with quantitative mass spectrometry that using a synthetic internal standard called QCOMCAT, and in collaboration with the laboratory of Brian Chait with Wensu Sang, we were able to basically get a very accurate quantification of using mass spectrometry of the relative ratios of every single nucleoporine that forms the core scaffold of the NPC. We not only that, but also with Erika Jacobs, we were able to get using label-free mass spectrometry, again, to verify the data that we were collecting using quantitative mass spectrometry, but also to quantify the amount of all the other transfer factors, flux, and material that was co-purifying with our NPCs. Next, in collaboration with Jennifer Gerton's lab, we use quantitative fluorescence to get the absolute copy number for several nucleoporins. And in collaboration with Martin Gerald and in their lab, using their setup for a charge detection mass spectrometry, we were able to actually fly whole NPCs in a mass spectrometer and get a mass for the whole assembly. Altogether, all this data actually were very consistent with each other and allowed us to define the size of the Saccharomyces cerevisiae nucleopore complex as being around 52 megadalton, and containing a surprisingly huge amount of co-purified material, 26 megadalton worth of transport factors, flux, and other associated complexes to the NPC. Next, to get the connectivity of the different parts of the nucleoporins of the NPC, we use, again, in collaboration with the laboratory of Brian Chait, Yishi, and Junji Wang, we use crosslinking, chemical crosslinking with mass spectrometry readout. That consists of basically using a crosslinker, a chemical ruler. A crosslinker is a chemical that has an arm of very defined length with two reactive groups on both ends of those arms. These reactive groups can basically react with two amino acids, and these species that are then generated between the amino acid, the crosslinker, and the other amino acid can be identified by mass spectrometry, and this will tell us that these two amino acids are within this distance within the complex, within the native complex that we are analyzing. In the case of the NPC, and because we had this new affinity purified prep, very efficient one, we were able to take this type of analysis to a whole new level and gather more than 3,000 unique crosslinks that actually cover basically all the different components of the nucleopore complex. And you can see here also that when we map these crosslinks later to the structure I'll show you before, all of them are within, are satisfied. They are within the range that not all of them, but a huge majority of them were basically validated and satisfied, and the distance between these amino acids that we were targeting in the different nucleoporins. Finally, to get the shape and dimensions and the fine detail shape of the nucleopore complex, we used a two-pronged approach. In principle, we use cryo-electron tomography to get a map, to generate a map of the isolated NPC. But then we decided to also use a two-pronged approach that should get the best of both worlds. And I'm going to explain that. We used our affinity isolated NPCs in collaboration with the laboratory of Chris Akey, to get a single particle cryo-electron microscopy map of the assembly. And this single particle map is going to have several advantages, because with the isolated NPCs, we can get really high particle numbers that will allow us to get really high resolution in many parts, the most conserved parts of the NPC. However, it has some drawbacks. For example, obviously, these NPCs have been isolated, extracted from the cell. So you lose the context, you lose their native context, and they are probably, we are looking probably at the structure of an inactive NPC, an NPC that is not actively transported. We think they are competent, but they are not actively transporting stuff. On the other hand, we decided to look at the in situ NPC by using a method that is called focused ion beam and cryo-electron tomography. And this was done in collaboration with Dynabeads in Elizabeth Villa's lab at University of San Diego. And in this case, we are going to have something quite complementary to the single particle EM. In this case, and because of the limitations of this method, we will have a low particle number. So the resolution that we expect is not going to be as high as with single particle. But however, we are going to keep the cellular context of the NPCs, and we are going to be looking at the structure of an actively transporting NPC in a cell. So just to show you, basically, because I think it's really beautiful, this is the schematics of the focused ion beam and cryo-electron tomography that Dynabeads did. So you can see here a pool of yeast cells, and they have been carved using a focused ion beam, gallium ion beam, to leave a very thin lamella of cells. You can see here, this is the lamella, these are different cells. And when we are lucky enough, we actually target a nucleus. And then in those nucleus, this is the nuclear envelope membranes, we can see here, here, and here, nuclear pore complexes that can then be analyzed by cryo-electron tomography. So again, these two maps will be highly complementary and informing each other about different aspects of the structure of the NPC. Here you have a picture of the final maps. This is the single particle cryo-electron microscopy map that gave us amazing resolution, close to 6 angstrom. Basically, we were able to see the secondary structure of most of the components of the NPC. And we also got a very beautiful in situ map that is informing us about other aspects and other parts of the NPC that were not so well resolved in the single particle. So all this data, together with even all the, much more data that we collected and that were also publicly available from other people and also from other labs. Again, we applied the integrative approach. We used all these different types of experimental data that we were able to collect to feed the calculation, translate them into spatial restraints, calculate and assemble a structure for the NPC. We then analyzed them, validated them through different types of criteria, and finally obtained a structure of the NPC that is a structure that has around 6 angstrom precision in some parts of the NPC. It's really good. I'm showing you this very brief video so that you can see all the different parts of the NPC, the outer rings, the membrane ring, the inner ring, the central channel. And here you have the blueprint for all the 550 proteins that form this massive complex. We can now show you how each of the different proteins connect to form subcomplexes, how they form the spokes, how they now connect to form the different rings and the different parts of the nuclear complex. And this was great. We have been able to get many different insights, but our idea was, okay, yeah, the structure is great, but what is really interesting for us as biologists is what is this telling us about the function of the NPC? What is the structure telling us about how the NPC performs its roles, and why it's built the way it is? And especially, in my case, I was very interested in knowing how the NPC can face, how the structure is designed or can help the NPC to face the different challenges that this molecular machine has to face in the cell. And those challenges are mainly two. One is the massive transfer flux that goes through all these NPCs at all times, as I showed you before, is really big and actually is all the time also regulated. And the other one is the tensions due to the nuclear envelope dynamics. Like, for example, as you can see here in this beautiful example of an anaphase bridge, even in yeast that has a closed mitosis, the nuclear envelope is stretched before the fission of the two nuclei. And there's a lot of different type of dynamics going on the nuclear envelope. And the NPCs have to be able to withhold those type of stresses and keep this massive flux regulated and going through them at all times. So the insights that we got as to how the NPC can face these challenges are several, and the first one of them is when we look at just how the NPC interacts with the nuclear envelope membrane. And when we look at this, the first insight was that the main component of the membrane ring, that's COM152, that has this Perlon string shape, forms and is arranged in a luminal belt that seems to be surrounding the core scaffold of the NPC and potentially forming a belt that delimits the level of dilation of the nucleopore complex. Next, when we also map how the different nucleoporins contact the nuclear envelope membrane, what we found is that they don't seem to be randomly distributed, but they form patterns like this this equatorial anchoring ring that seems to be forming what we call a firm girdle at the level of maximum tension of the nuclear envelope membrane and stabilizing that region of the membrane. Then also, when we look at how more peripheral proteins connected to the membrane ring, we found that they seem to be forming this, guiding the curvature of the two leaflets of the two parts of the nuclear envelope membrane, the inner and the outer and the inner, and forming what we call clamps that seems to be stabilizing this type of curvature that the NPC has in the nuclear envelope. So all this combination of luminal belt, girdle, and clamp is what seems to be allowing the NPC to generate the channel to stabilize and curve all these membranes of the nuclear envelope. Next, and in collaboration with John H.'s lab using a phenotypic assay, we were able to identify the presence of relatively rigid diagonal columns at the heart of each of the spokes of the NPC. These columns provide some rigidity to the spoke and also can potentially move and to accommodate expansion and compression stresses. Because each of the spokes, however, although they are relatively rigid due to these rigid columns, however, when we look at how they connect to each other, they seem to be connected with what we call hinge connections. These are very weak or relatively weak and not very extensive connections between the different spokes that form the NPC, so that actually it allows probably each of the spokes to move independently from each other. And this is very remarkable, and it was quite clear when we look and compare the structures of the isolated NPCs and the in situ NPCs. When we look at those two different structures, two different maps, cryo-EM maps, we saw that the isolated NPCs seem to be adopting a constricted structure where each of the spokes is really close to each other, establishing these hinge connections. But when the NPC is in an active state in situ, with also keeping the tension of the nuclear envelope, these spokes separate from each other. They don't seem to change massively in their conformation. It's just that these hinge connections are now lost, most of them. You just have a couple of them to keep them connected. and generate these gaps in the structure. You can also see this in this video comparing the constricted and dilated version or architectures of the nucleopore complex. And just to show you in a more diagrammatic way, what we can see is between the active and the constricted NPC, we see that the POMA52 luminal belt seem to be expanded and defining the maximum level of dilation of the NPC. And each of the spokes, they don't change massively in their conformation. What is going on is that actually they separate from each other, leaving these gaps that we think are also and also expanding the diameter of the central channel. What we think might have implication for several transfer pathways that go through the NPC. And this is thanks to all these hinge connections, and also thanks to another feature that is very important in the NPC that I have not mentioned before, and those are the flexible connectors. And these flexible connectors are, again, something that was first described in this groundbreaking paper by the Ed Hertz lab. They are relatively long, intrinsically disordered regions in certain nucleoporins that contain short interaction motifs that are going to start establishing weak interactions with some of the folded big components and big modules of the nucleopore complex. So you can think of them as a piece of string with a couple of pieces of tape that then you use to connect certain things. But what we can now see in the structure is that these flexible connectors are actually really important, and they form a whole network that interconnects all the different rigid modules of the NPC. What we see is that the flexible connectors thread all the way through from the cytoplasm all the way through the spoke and to the nuclear side where the nuclear basket is, connecting all the different modules of the NPC. And they also thread radially from connecting from the nuclear envelope membrane through the core scaffold of the NPC and even into the central transporter in the central channel of the NPC. So we have all this network of what you could consider flexible cables connecting the different rigid modules, providing flexibility and resilience to the structure of the NPC. And the final insight that biological insight that I would like to share with you is came a little bit as a surprise, and it basically addressed a question that we have been asked many times when we talk about the nucleopore complex, and that is, are all the NPCs within a same nucleus the same? Because we know that if we look at different type of organisms, the NPCs have some certain diversity in their architecture, especially in the outer rings and the peripheral components. What we have seen is that, for example, in yeast, it was described that there are single outer rings. In, for example, an algae, like we see a single outer ring in the cytoplasmic side, double outer ring in the nuclear side, while, for example, in invertebrate, in humans, we have double rings in both sides of the NPC. So there's an architectural diversity in between organisms, but it was normally accepted that within the same cell, all the NPCs were probably very similar. And in this case, what was a big surprise and came out of the purification of nucleopore complexes that we were able to design and use, came this surprise that I was telling you, because when we actually analyzed, when Chris Akey analyzed the data set that we collected in the isolated nucleopore complexes by single particle electron microscopy, he did a much in-depth classification and multipodal refinement. What we found is that in comparison with the in situ map, where we found the canonical single ring in both sides of the nucleopore complex in yeast, we found that actually a percentage of the NPCs in yeast show a double ring in the nuclear side of the NPC. We estimate this to be around 20% or so. And this was validated in collaboration with the Stowers people, Sue Jespersen lab, by fluorescent, in vivo fluorescent microscopy. And what we were able to basically get now a nice structure of this double ring that is in certain ways similar to the one that has been found in vertebrates. And in other cases, some of the components are doing something similar, but in a completely different way. This component in the human part are located on the other side of this double ring. Here they are on the opposite side. So it seems like during evolution, eukaryotes have been able to use the same components in a Lego-like fashion to actually build alternative architectures in the NPC. So I mean, I'm not going to go into deep details here, but just the take-home message is that we have been able to now find at least 3 different isoforms of nucleopore complexes coexisting in the same nuclei. Basically, a form that is more the canonical NPC with single rings and a nuclear basket. We found the one that has a double ring in the nuclear side and also nuclear basket. And surrounding the nucleolus, we also know it was described before, that is mainly a single ring and no basket type of NPC localized around the region of the nucleolus that is pretty big in the yeast Saccharomyces cerevisiae. So the presence of these different isoforms opens very interesting questions, because we think that these alternative forms of the NPC might have also potentially alternative functional outcomes. They might be involved in either transporting different types of things or performing different regulatory roles in these cells. So this is opening a whole new avenue of investigation that we are really keen on pursuing in the future. And yeah, so just to summarize what we have learned about the structure of the NPC, I would like to use, we like to use an analogy, and that is that the structure of the NPC is analogous to the one that of a suspension bridge. In both an NPC and a suspension bridge, they are structured that are designed to connect 2 parts and allow a massive and constant flux going through them at all times. In both suspension bridge and the NPC, we have firm anchors that to the substrate, in this case, in the case of the NPC, to the nuclear envelope membrane. In the case of the bridge, to the bedrock. In both of them, we also have relatively rigid modules that are connected through flexible hinges so that they can move independently from each other, and connecting all this whole network of flexible cables that connect the different rigid modules of the structure. What you end up with this is a very strong yet flexible yet resilient structure that can withstand many different types of stresses. Both also in the NPC, what we have found is that it is not only that flexibility goes into the structure and the conformation, but also in the conformation and in their composition, potentially generating a different type of architectural isoforms of the NPC. We know there are differences in different organisms. Now we are seeing that it is potentially also even within cells, this diversity. And just to finish, I would like to just summarize that I think I've shown you that we have been able to generate this optimized methods for the isolation of intact native yeast NPCs. This method we are convinced that is applicable to other type of large macromolecular complexes, or even to other type of organisms. And at this point, I would strongly encourage any of you that is interested in this method or similar methods to visit the website of the National Center for the Dynamic Interactive Research, NCDIR. It's a consortium of laboratories funded by the NIH in the National Center. We are part of this. And in the protocol sections, you're going to have all sort of very detailed and very useful protocols, from conjugation of Dynabeads to protein complex purification, and many others that I have not even mentioned. We also have video protocols, video resources, and all these protocols and video resources are we keep adding them as we develop them. So I'll strongly encourage you to visit this website if you want to get some of these protocols, or also directly contact us if you have specific questions or you have any specific issues with some of these methods. Again, we think that these purifications are ideally combined with integrative approaches if what you want is to analyze or solve the structure of and characterize the structure of these complexes. In the case of the NPC, we have actually found through solving this structure that is a finely tuned biological machine based on very sophisticated biophysical and engineering principles. And that actually, it seems to have an unexpected structural, potentially also functional diversity that we are very excited about and that we would like to explore in the future. And with that, I would like to basically thank everybody that was involved in all these projects, all my many and amazing collaborators in Rockefeller, Stowers, Chris Akey, Elizabeth Villa, Ishi, John Aitson, Andre Salis, Steve Lotke, and Martin Jarrell, and all the people that also the funding agencies that gave us the money to do all the stuff that we do. I would like to thank you all for your attention, and I'll be happy to take any questions that may arise during the talk. So thank you so much.
Okay, thank you very much, Javi, for a fantastic talk. And we have several questions in the Q&A, so I'll start with the first one right at the top. Someone is asking, they're anonymous, so I don't know who they are. What cocktail of protease inhibitors were necessary with or without mutations in the corresponding yeast genes to suppress the actions of the endogenous yeast proteases to avoid damage?
Mm-hmm. Yeah, in our case, we are using the protein inhibitor cocktail from Merck. There are many different types of commercially available. This is a complex mixture. We tested different ones. We were also using a mixture of peptidin A and PMSF. We call that solution P. But in our hands, to be honest, these cocktails have been performing really well. So I would suggest this one from Merck is a mammalian protein inhibitor cocktail. And also we have used the ETA-free little tablets. They also work really well. And we have actually got pretty good inhibition of degradation, both in yeast, in trypanosomes, and also in mammalian cells. So you don't need to make your own very complicated mixture. Some of the commercially available are perfectly fine and are most of the time gonna do their job. You can look at their composition in their websites, but yeah, they're complex cocktails that will allow you to prevent degradation from many different types of proteases.
Okay, I have a question from Arco that says, if you use antibodies for affinity purification, where would you need the HRV protease exactly?
The HR what? Sorry?
I think they mean the PPX, why you need PPX cleavage if you're using antibodies.
Well, it depends. The point of the cleavage, of the protease cleavage, is to really cut the connection between the handle, the tank that you're using, and your protein, so that you can release your complex under native conditions. Obviously, for example, if you're using a flag antibody, you can use the flag peptide to perform the same type of native elution. If you're using, I don't know, strep tag, you can potentially use biotin. There are different methods to do this. In our case, and for different reasons, the one that we chose, and because it's easy and inexpensive to genetically modify yeast, we introduced this cleavage site, this product cleavage site in between the tag and the protein of interest, and it has performed really well in our hands. But again, there are many different options, and you just have to look at the one that basically suits your downstream interests, and that is the best for your complex.
Okay, there's a question from David. How many cells do you need as starting material, assuming the usual yield for mass spec analysis of NPCs?
For mass spec, it depends what you want to do, but for a mass spec sample, I would say that around 1 gram of wet cells. How many cells is that? I will need to do some back of the envelope calculation, but yeah, that is what you might get out of growing around 200, 250 milliliters of mid-log phase yeast culture. So yeah, you're going to have 3 times to the 7 cells per milliliter. So yeah, you can do the math there. But that's what I think it will be enough to get nuclear pore complexes using our methods. 1 gram of wet material will give you enough NPCs, around 5 micrograms, to run even quantitative mass spectrometry of label-free mass spectrometry. If what you want is just IDs, much less. I mean, native mass spectrometry nowadays is super sensitive, so a small fraction of that will give you enough to just get the IDs. But if you want to be quantitative, you need a little bit more material.
Okay, thanks, Javi. There's a question from Althea. They're asking, do you have to tag the protein? Do you have to tag the protein at the yeast genomic level, or provide a plasmid with the tag component of the complex?
You could do both. Yeah, both should work. In my case, I usually prefer to genomically tag them, because it's not that difficult, and you ensure by doing that your protein is expressed under the control of its native promoter in its native locus. So you try to prevent introducing more changes into this strain. Adding a tag is already a mutant, so you have to be also very careful. Whenever you tag something, you have to double check that it's viable. You don't see any obvious or rough phenotype showing up, and that your protein is active, is incorporating where it should. So you require a little bit of work before doing that. But once you have seen that, I don't think it should be a problem to use either genomically tagged or a protein that is tagged and is expressed from, let's say, a centromeric plasmid. Both should work absolutely fine. In the case of the genomic tag, if you're using a haploid strain, all the copies of your protein are going to be tagged. If you use a diploid with only one of the copies, you're going to have a mixture. And in the case of a plasmid, you usually will have a mixture unless you're using the plasmid in a background where you have a deletion of your complex. So that's another thing that you might want to consider. If you want all the copies of that protein of interest being tagged, or you also want a mixture of tagged and untagged proteins in your complexes.
Okay, cool. No, thanks. There's a question from Misha at NYSBC. What is the concentration you typically obtain in your purification, and what would be ideal for the cryo-EM analysis that you showed?
The concentration, I don't remember from the top of my head in terms of molarity. I can look for that and tell him. Usually we are getting around 0.3 milligrams per mil of protein in our final preps, and that's usually the type of prep that has been ideal for cryo-electron microscopy. However, I have to say that it's not really a matter of the concentration of the sample. To get good grades for cryo, you need a lot more, a lot of other things, and it requires a little bit more treatment. You have to incubate them the right amount of time, the right amount of grids, doing the right amount of washes, and with specific buffers. And then obviously, and he knows much better than me, to be able to get proper eyes and well distributed. In the case of the NPC, for example, you also need grids with that are carbon coated, because they don't like holy grids where there's nothing that they can attach to. They will immediately tend to aggregate on the sides of your grids and basically be unusable. But yeah, that's usually the amount of material that we use.
Thanks. Question from DP Varma: is any component of the NPC glycosylated?
It's glycosylated. There are glycosylations in the NPC. I think some membrane components and even certain FG nucleoporins have been shown to have a specific type of glycosylation. But yeah, something that I'm not very familiar with. But I know they are. We have not analyzed if we are keeping or maintaining that type of post-translational modification. It will be interesting to look for that and see if it has any effect on the structure. But we have not. We are still not yet there. So yeah.
Yeah, I think there may be some in mammalian FG naps, but I think I'm just remembering off the top of my head. Okay, question from Lynn. Which chemical crosslinkers were used? Sorry, this didn't jump. Which chemical crosslinker were used in the NPC complex identification?
Sure.
How about the effect of different linker? Okay, do different linkers provide different results?
Yes. So we mainly use two different type of crosslinkers. We use crosslinker that is called BSS. The succinimidyl suberate has an arm of around 13 angstrom, and I think that the reach with the side chains gets to like 20 something, between 24 and 30 angstrom. Distance is the type of restraint that we apply to those. And this is homobifunctional and will react with primary amines. And then we also use EDC, that is a close to zero distance crosslinker that actually is heterobifunctional. It reacts with primary amines and carboxylic acids. What we found is that, interestingly, well, we and others, I know that the group of Rudy Aerosol and others have shown something similar, is usually is more interesting to have complementary chemistries than complementary distances for the crosslinkers. You will get a more complementary data sets if you can combine chemically different crosslinkers, that if you just combine a crosslinker that is 13 angstrom reach with one that is 4 angstrom reach, but has very similar chemistries, like, for example, amine reactive. Your data sets are going to be quite overlapping in the end. So yeah, if you can, and if you have the possibility, I would strongly recommend having complementary chemistries in the crosslinkers.
Thank you. Question from David: can we use a more aggressive methods if our object of study is more focused in a specific part of the NPC, for example, the basket, for biochemical characterization rather than the whole complex structural studies?
Yeah, of course, you can. We've done it. For example, at some point, we to analyze some of the subcomplexes of the NPC, like the Y-shaped complex that forms the outer rings, or the NuPETi2 complex that is forming the mRNA export platform in the cytoplasmic side. We use much more aggressive extraction methods that basically, blow apart the NPC, but keeps these other subcomplexes stable and happy, and we can purify them. So in terms of the basket, sure, you can. I'm sure there are conditions that would be suitable for separating the basket from the NPC and isolating it. In the case of the basket, in my experience, the only thing I can say is that it's quite challenging to do that. But I'm sure there's somebody that is going to be able to do it one day. So maybe that's him. In any case, yes, if you use harsher conditions, you will break apart the NPC, but that will allow you to specifically potentially purify some of the subcomplexes and components of the NPC.
Okay, I have a question from an anonymous person. Has the structure of the NPCs of cancer cells been studied and compared to NPCs of normal cells?
Cancer cells. There are a couple of papers where they have looked at cancer cell lines. The problem is that normal cells is a complicated term because most of the analyses that I know of have been done in cell lines. Cell lines, I would not consider them normal cells because they are not. They're transformed, immortalized. If you think about it, they are kind of like little monsters. So I'm not sure how good that type of comparison is going to be. There are papers, I think in the paper from last paper from Tonsworth with Mary Dasso and Hadmedalia, they looked at some cancer cell lines. I know that also Marinberg looked at the composition differences in the composition of different cancer cell lines, and there seem to be differences, but I am not sure if it has been the NPC of a normal cell, whatever you define that, has been solved. So it would be something interesting to look for. Potentially, it's going to be very similar to the maps that we already have from cell lines, but that would be an open question.
Thanks, Javi. There is a question from Tony Isebe, a graduate student from McGill University, asking to check the connectivity of nucleoporeins to the NPC via crosslinking. Do you do it in vivo or in vitro? And if in vivo, how do you control for non-specific binding where you have the crosslinker binding to largely abundant proteins?
Mm-hmm. So the type of cross-linking mass spectrometry data that I've shown was done ex vivo, in vitro. It was done on isolated, purified, intact NPCs. The type of cross-linking in vivo that he's describing is something that we are very interested in pursuing. We are actively looking for ways to do that, but it's quite challenging right now, especially because of the type of issues that are mentioned in his question. It's potentially difficult to find an equilibrium between getting good cross-linking, good coverage for your proteins, and not starting to cross-link everything else that is around the cell. So we have not been able to do cross-linking with mass spectrometry readout from samples that are in vivo yet.
Okay, thanks. There's a question from Song Hao. Is it possible? Sorry, this thing there. Is it possible to use other affinity tag peptides? Are there any recommended choices, and can the Dynabeads be repeatedly used? I think that's multiple questions, actually. So is it possible to use other affinity tag peptides? Are there any recommended choices, and can Dynabeads be used repeatedly?
So Dynabeads can be used repeatedly. I would not recommend it if your downstream application is very sensitive, because it might be, you can wash them and strip them of everything, but your efficiency, your pull-out efficiency is going to drop significantly depending on the treatment. So you can reuse them. We usually just reuse them for like performing tests. If you're going to run an experiment that actually you expect to give you good data, I would not recommend to reuse Dynabeads. And in terms of different type of peptide handles and tags, yes, you can use different ones. In our hands, GFP has performed really well and has the advantage of being able to also do some cell biology. So all the fluorescence tags work really well. The flag tag, several people in our lab has been very successful in purifying big complexes. We have not tested it with the NPC in particular. Other tags in our hands were not so great, but that could be due to many different reasons. So I'm not going to tell you, oh, this tag is not good. That's not the case. It was potentially not good in our conditions for this specific complex, but potentially if it binds with high affinity, you can natively elute it. It should be possible to use, yeah.
Okay. Another question, if you did not include any phosphatase inhibitor during the prep, did you see any significant difference or change in the NPC structure compared to when you did have a cocktail of phosphatase inhibitors present during the prep? In other words, any evidence at all that phosphorylation has any effect on the NPC structure?
We know phosphorylation has effects in the NPC function. We don't know if phosphorylation has effects in the NPC structure. We have not tested that. We have not done a comparison between a fully phosphorylated and a fully dephosphorylated NPC. That's something that potentially we will be able to do in the future. In my hands, we didn't see obvious changes in our preps when we use phosphatase inhibitors or not. So at some point, and for the sake of simplicity, we didn't include them. But definitely phosphorylation is one of the modifications that help modulate the function of the NPC. So in that sense, yes, that would be something interesting to look for. I don't know if it's going to generate structural changes at a level that you can detect with the methods that we are using, but it's definitely something that would be interesting to look for in the future.
Another question from Sung Hao, is there any protocol to get the pure genome edited haploid strains? If the genome editing leads to some hybrid diploids, I'm not sure I followed the question, but I'll just repeat it again. Is there any protocol to get the pure genome edited haploid strains if the genome editing leads to some hybrid diploids?
I mean, in yeast, and that's not our protocol or anything like that. There's decades of genetics, and you can go online or any handbook in yeast genetics and yeast manipulation will show you that in a haploid yeast, you can quite easily genetically modify them. And then there are different methods that you can use to actually look that your mutants are clean. I don't know, on top of my head, the most kind of like in-house or easy to do one would be with a southern blot or diagnostic PCRs. There are more sophisticated ones where you look at to see if there's not been any type of chromosomal duplication or stuff like that. It also depends on what you want to do in your downstream applications for these strains. But there are definitely methods. And I cannot point you exactly to a method or a paper, but I'm sure you can easily find it, because there's a lot of literature in this topic.
Okay, question from Ramya. Can NPCs be regulated by drugs? Can we stop certain signal pathways that trigger apoptosis or cell division so that we can use them as therapy for cancer?
There are pathways that can be chemically modified, and actually relatively recently there's 2 drugs, I think, that have been approved by FDA to treat cancer, that actually target specifically one transfer pathway, actually the export pathway of CREM1, that is selinexor and verdinexor, I think they're called, these drugs. So yeah, it's not only possible, it is happening. In terms of blocking apoptosis, I cannot tell you right now. I'm not familiar with apoptosis, but it is possible to block specific or to use drugs to affect specific nuclear transfer pathways. So if some of the aspects that you want to modulate are going through a specific transfer pathway, you can potentially find drugs that will target those. So yeah, I hope that's an answer to that question.
I think there's 2 more questions. One is from Bhavesh: how to co-express 2 interacting proteins of the NPC without using any linker between the 2 in a yeast cell?
How to co-express 2 interacting proteins? Well, you can co-express them in any system, recombinantly or in yeast. If they interact, they should do what they do, that is interact with each other. I mean, I will probably need a little detail in terms of what he wanted to do, he or she, but yeah, it is totally possible. Yeah, you can do that.
Okay. Stephen Sakuma is asking, what modifications would be necessary to adapt this to mammalian cell lines or tissues? Do you know of anyone who is planning to try this?
We have tried it, not very successfully, and I'm pretty sure that there's people out there trying it. Again, if I knew, I would tell you, but I don't know. I'm sure there's, starting from the type of material, the type of cell line, I know there are mammalian cells that don't have very strong lamina. That might be a way to start with. And then again, you should probably also spend some time and effort on optimizing buffer conditions. What we see is that if we just take our protocol for yeast and apply it to a mammalian cell, it doesn't work very well. So yeah, there's a lot of work that will need to be done there to optimize the different parts of the protocol, different conditions, to be able to purify an NPCs, same way we're doing it with yeast for mammalian cells. That said, I'm absolutely sure that it can be done. You just have to find the right conditions.
Okay, final one is Rakesh wants to know how you got an in situ model of the NPC, basically, as opposed to isolated. How do you get the in situ?
Well, as I showed, it was by using native cells that are growing, they are frozen. Then we use a focus ion beam to carve thin lamella that basically is like a section of those cells. And then using cryo-electron tomography, Dick Vijay Singh in Elizabeth Villa's lab UC San Diego, were able to collect enough data, enough tomograms of NPCs to reconstruct a map of the in situ nucleopore complex in an actively growing yeast. So that's how it was done.
Hey,
All right.
thank you very much, Javi. That was quite a few questions actually, and lots of comments in the chat about what a great talk it is, and everyone is grateful to have learned from your methods. Thank you.
Well, really, thank you so much to everybody that attended, and thank you for the comments and the questions. I hope it was helpful. And yeah, and in any case, there are in the website and bioprotocol, there's a section also where you can post any other question that you have. I'll try to answer them later or some other time. And yeah, we are always open for anybody that wants to contact us with question details or whatever.
All right. Well, since there are no more questions, we will end here. Thank you so much for this interesting talk, Javier, and this lively discussion both in the chat and at the end of the session. I think we had sufficient time to address all the questions, and I hope this was helpful for everyone. Thank you. And as Javier already mentioned, if you have more questions, please feel free to visit our website, where there's a Q&A section where you can still post questions after the webinar or participate in a discussion if you feel you can answer a question. As I mentioned earlier, the webinar is being recorded and will be posted on our website later. And this might take about a week or two until it is uploaded. And lastly, I would like to announce our next webinar, Beyond NGS, Single Molecule Genomics by Yuval Ebenstein, moderated by Alessandro Di Donna, on March 7th. And if you want to know more about this webinar, please visit our website. Thank you again, everyone, for joining today, and hope to see you next time.